Hello, good day, friends. It's wonderful to have you all together again this evening. And this evening, we'll continue our discuss. We've been discussing immunology and the immune system. So, this evening, we'll continue by discussing a few concepts in immunology and about immunity so that we get better understanding about immunity, what it is, what it entails, and everything. Today, we'll be discussing about we'll be discussing, like I said, some concepts. We'll start by discussing active and passive immunity. What's active and what's passive immunity? Active immunity talks about immunity that is conferred on an individual on the basis of exposure. Before the individual can say that he has immunity to a particular disease, pathogen, or infection, that individual has been exposed to that particular pathogen, illness, or infection. And on exposure, the human body, or the immune system within the human body begins to build resistance, begins to build strength and resistance against this foreign pathogen. And when this kind of immunity is built, we refer to this kind of immunity as active immunity. It is active because it is based on the fact that the individual involved has been exposed, actively exposed to this illness. So if you have a baby, for an example, taking vaccination, what you are essentially doing by vaccinating a baby is exposing that baby to different pathogens, exposing him to tuberculosis, exposing him to diphtheria, exposing him to cholera toxins, or whatever the vaccine may be. So within the baby, you are trying to build active immunity, and that's what active immunity is immunity that is conferred on an individual on the basis of exposure. Now, apart from active immunity, we also have what we refer to as passive immunity. And passive immunity is a situation where there is immunity conferred on the individual, but it's not conferred on the basis of exposure. It's not because the individual has been exposed to the pathogen. Rather, it's a situation in which a parent organism or an organism has been exposed and has generated or gathered antibodies against that pathogen, and these antibodies are transferred to another organism and they provide protection for that organism. The simplest situation where you have this happening is a baby in the womb of the mother. When the baby is in the womb of the mother, it's a simple situation. The mother can get exposed to different kinds of pathogens, different kinds of organisms, different kind of illnesses. And when the mother gets exposed, she builds resistance in the form of antibodies. And when these antibodies, or immunoglobulins as we like to call them, are built, they can pass, some of them like immunoglobulin G has the capacity to pass through the placenta of the mother to the child, and then it provides protection for the child. So this is important. This is important to note. That's what passive immunity is about. So active immunity, the organism, the baby or the child himself is actively exposed to the pathogen, and then there is the generation of immunity and protection for the child. But in passive immunity, it is the mother or another organism that enjoys that exposure, but the antibodies that are produced to protect that mother, or that mother, and is now passed through the placenta or through some other means to provide a general protection for the child. So that's what we refer to when we're talking about active and passive immunity. Now, we also want to talk about antigens. What are antigens? Antigens are organisms that are able to elicit or trigger an immune response within the human body. So any organism, any pathogen that finds its way into the human body, and when it gets into the human body, it is able to elicit, it's able to generate, it's able to provoke an immune response from the human body. That kind of organism is referred to as an antigen. So antigens are very important because they play a role in, once they come into the human body, they must be recognized, and when they are recognized, the immune system must find a way to get rid of them. There are many ways through which antigens can find their way into the human body. They can be ingested along with our food. That's if you drink water or you eat food, you can swallow pathogens, swallow antigens. You can use the terms interchangeably. Pathogens, antigens, you're talking about the same thing. Once they are able to elicit immune response within the body, we say they are antigens. Sometimes we take it to the level of saying they are immunogens. Now, apart from that, is either you ingest them, you can get them through an injection through the skin. An insect bite or something that is able to pierce through the skin can putting antigen into the body. There are other means also, sexually transmitted. If somebody engages in risky sexual behavior that is not protected, such an individual can have antigens finding their way into the human body through any of those means. And this is important for us to know. Now, there's also a term that is important for us to know. There's what we call antigenic determinants. Antigenic determinants. And another term for antigenic determinants are epitopes. And what this simply means is that there are molecules that are present on the surfaces of antigens, and it's these molecules that help the antigens to trigger an immune response. It is also to these molecules that antibodies that are produced by the human body or the human immune system in response to the presence of these antigens, they attach to the antigens on these sites, the sites of these antigenic determinants or epitopes. So it's important to know them, and it's important to know what they do. I said they are molecules, can be proteins or lipids or even carbohydrates or glycoproteins, and they are present on the surfaces of pathogens. And that their presence is important because it is through is by using them one is able to attach the antibodies to the antigens. So this is important to know. It's also important to know that there are about 3 types of antigens that we talk about. We talk about autoantigens. Every cell within the human body has its own antigens, and they are molecules through which the immune system is able to recognize cells, and so it doesn't fight against itself except in the advent of an autoimmune disease. So you have autoantigens, which are the antigens of the human body itself, which the immune system should not fight against. You also have alloantigens. These are antigens that are sometimes found on antigens that are found on blood cells, red blood cells. When you say somebody is A positive or A negative, rhesus factor, all these kind of antigens are what we refer to as our alloantigens. Then you have what we refer to as our heterophile antigens. These are antigens that are coming from outside. Now we'll wrap up this small discourse by talking about antibodies. Antibodies, another name for antibodies are immunoglobulins, and these are proteins that are produced by the human body in response to the presence of antigens. They are produced, there are many theories, but certainly for now, we use the monoclonal antibody theory to explain the production of these antibodies. They are generated in response to the presence of an antigen. So if an antigen finds its way into the human body, the immune system wants to produce antibodies en masse to be able to combat these antigens, attach themselves to these antigens, and over time, with the help of T cells and B cells, destroy these antigens. These concepts are important. We'll continue our discourse on immunology in the very, very next class. Thank you for listening. Remember to subscribe, like our content, subscribe, and do have a wonderful day.